Okay, welcome to my channel. Now, as we have already in the last video, we have posted the video of the cell theory and the cell structure. Now, this is the video, as I told, that next video will be on animal cell, a typical animal cell. Now, as we have talked about that there will be two types of cell categories we will be studying out. The first one was the animal cell, and another video which I will upload is on the plant cell. So about a typical animal cell. Now there are different, you can see in this board we have already had a drawing in order to consume our time. So I have already have a rough drawing of a typical animal cell in the board. Okay. Now in this particular stage, we have seen students drawing different types of extra structures. They go for extra perfection in doing the drawing. No need of that one. In case of a typical animal cell, we desire the varieties of things which is with their specific function. We want those things. Okay. So for a simple diagram, this is a typical animal cell which is being shown out here. Now this is a outer membrane or the cell membrane. Now as when you are doing a drawing, suppose if in case of the ICSE, CBSE, especially they give you the drawing and they give you the numbers to point out what is the cell organelle. In that case, it is okay that you are only pointing out that what is which organelle is which one. But in case of the descriptive board like the central means the state board, sometimes they say that draw a typical animal cell. So in that case, these are the things which is desired. Okay, the first thing what you will draw is the cell membrane. So this is the first one which is going to be drawn outside the animal cell. This is the cell membrane. Now cell membrane is also called the plasma membrane. Cell membrane is also known as the plasma membrane, or it is also called the semi-permeable membrane. So first thing what we start off in animal cell is a plasma membrane. Now conceptually, what is a membrane? A membrane is a structure that is a boundary. So in order to perform different types of activities, we need a boundary, we need a limitation to everything. So such like that, a cell is actually covered by a cell membrane or a boundary, a limit. Okay, so this is what is a boundary or the cell membrane. Now this is also known as the plasma membrane or the semi-permeable membrane. Why is it semi-permeable? Now the word semi and permeable. Why permeable? Why semi-permeable? This is also called a semi-permeable. The word itself, permission means what? Giving allowance to someone, right? So semi means not fully. It's partially allowing certain substance to go inside the cell, and it is restricting certain toxic substance to go inside the cell. So this is the boundary. This is porous in nature. Porous means it has pores in it that will allow certain substance to go inside and will restrict the others from going inside it. So this is actually acting as a boundary, a protective cover, and it is giving shape to the cell also, and it is also allowing the permission of certain things to go inside and restricting the others to go inside or come outside. So this is basically the function of this cell membrane, is to provide protection. So the first thing, what is it? You in an animal cell is a cell membrane. In case of a cell membrane, there is nothing to get confused. Cell membrane is also called plasma membrane, or it is also called a plasma lemma, or it is called a semi-permeable. Why semi-permeable? Because it allows certain substance to go inside and restricts the other. So this is why it is called a semi-permeable membrane. Next, coming to that one is the first one we have done, cell membrane. Then we come to the cytoplasm, this plasm. Now if I break the word, that means if I break it into cyto and plasm. Now see, plasm means plasma, cyto means cell. If you read in this way, it will never get out of your mind. Cyto means cell, plasma means any jelly-like thick substance like jelly, like any viscous fluid, that is also called a plasma material. A plasma material actually provides the base to this whole cell so that other cell organelles can float inside it, and it maintains the sustenance of the cell. It actually maintains the balance of the cell, and it provides a medium. Now, if you put the cell organelles in the vacuole, will that function? No. So we need a base. So that is acting as a base. Cytoplasm actually acts as a base to provide these cell organelles to float inside it. So that is why the first thing is cell membrane, and then is the cytoplasm. How is the cytoplasm? We have in this diagram, no need to draw. Certain students draw it like a fluid like structures or sketch it dark. No need of that. We have to mark it as this dot 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 structures. This can easily identify that you are drawing the cytoplasm. Clear? So cyto means cell and plasma means the fluid material or the jelly-like substance. So together we call it as a cytoplasm. Function, it provides a base to all the other cell organelles to provide the function. Clear? I hope it's clear. After doing that one, cell membrane, cytoplasm, next we come to this very important unit, that is the mitochondria, or cells mitochondrion. Okay, so don't get confused with the names. The names itself gives the idea of the function. So when you are studying the name, you are getting half of the functions only. Mitos and the chondria. This is the unit. This unit, mitochondria. Actually, what is the function? It is a double layered structure. See, double layered will always be like this. So in a double layered structure, what is the main function? It is also called the powerhouse of the cell. Why? It is also called powerhouse. Often questions come that which is the powerhouse of a cell. Now please do make a conceptual allied thing clear. Powerhouse. What is the function of powerhouse, children? Powerhouse is that from where we get the energy, right? So powerhouse is the form where the energy is produced. Just like that, in this mitochondria, the powerhouse, it is the energy is produced for all the functions. We are doing different types of activities in our life. Now, these activities are actually generated where? In the mitochondria. Mitochondria gives the full energy for a cell to work, divide, and all the performing activities are actually controlled by this unit. This is a master unit, and this is the mitochondria. If we remove the mitochondria, conceptually think, children, you can make a comment box also. You can write me in that if we remove the mitochondria, will the cell function? Or what can be the fate of a cell if we remove this mitochondria? Clear? Next one. Next we come to the next part of it, the Golgi apparatus. You can eventually draw like this. Okay, Golgi apparatus are like this. These are the Golgi bodies. They are also present just above this one. And while student you are marking the diagram, it is a request to you, don't ever draw in this way that you are marking some of the organelles on this side and then you are making arrows on this side. No, you have to draw in one side only. If you suppose, just like see this one, when I wanted to point out this one, I have not allowed it here. What I have done, I have marked it, arrowed it, and then I am taking the stretch and I will give all the list of the organelles in one side. Never use this arrow, this arrow, this arrow. No, this is not done. You are not supposed to draw like this. Whatever you have to mark, take it in this way and mark it in one side. Either you can choose right or you can choose left as per your priority. Next one, we go to the After the mitochondria, we have discussed this is a Golgi apparatus. Now Golgi apparatus, the functions all you know. This is a type of a stored for that we have different types of organelles out here. Now see, after this one, after Golgi apparatus, we see this one. This is a small blank space. You can see here, you can see here. Now in case of animal cell, this vacuoles, these are called vacuoles. This is the main function of vacuoles, is to provide support to the cell, right? So vacuole in case of animal cell are very small, or sometimes it's negligible or it's not there. Vacuoles are actually produced during the need of the cell in case of animal cell, but in case of plant cell you will see vacuoles are playing the main part of the cell, but in case of animal cell these are produced as per the need of the animals. Okay, animal cell. This process of production of the vacuoles and again disappearance of this vacuole is called pinocytosis. Pinocytosis. Just make your notes. This is a process. Pinocytosis. Pinocytosis is a process in which what happens, whenever vacuoles are needed, it is produced. When it is not needed, it is being removed out. So this is the process which is called pinocytosis, right? So vacuoles' main function is to provide support whenever it is needed, but in animal cells it is sometimes present and sometimes it is not present also. So it's not a very vital thing for an animal cell. Next we come to these particular structures. Have you seen these particular structures? These are the ribosomes. Now ribosomes play a very important role. As you know that ribosomes are the main unit for protein synthesis. You will come when you are studying genetics or you are studying the extra level of the molecular biology, you will come to know the functions of ribosomes. It is, although it may be a very small structure, but still it plays a very vital role as it helps in protein synthesis. Protein is very much important. So synthesis of protein is the site. The site for synthesis of protein is ribosome in case of the cells. Okay, next after that one is this type of structures. Can you see this half broken structures? This is, and this one. Now the structures which are actually having ribosomes present on it, these structures, these half-life structures which are having ribosomes present on it, they are called the rough endoplasmic reticulum. Rough endoplasmic reticulum are comb-like structure. Have you seen a broken comb? It's just like a broken comb-like structure. So in case of endoplasmic reticulum can be of two types. Now see, if you write endoplasmic reticulum. Now see, this word when was written in a whole was giving a complex idea, but when I split this word, see, it's becoming easy. Endo means anything which is inside. Plasmic means plasma type, means the main material which is forming it is a plasma material or a viscous jelly like substance. Reticulum means what? A structure that is a reticular structure. A type, just I told you that it's like a broken comb, reticular structure. It's a type of reticular structure. So that is why it's called a endoplasmic reticulum. Now, endoplasmic reticulum can be of two types, right? What is it? One is the SER or the RER. Now often questions come, what is the function of SER or what is the function of RER? Now SER means smooth endoplasmic reticulum. What is it? This is the smooth endoplasmic reticulum. Means when the endoplasmic reticulum is just like this and there is no extra ribosomes attached to it, then it is called a smooth endoplasmic reticulum. But the moment endoplasmic reticulum having ribosomes present on it, that means the granular structures, the ribosomes are present on it, that time the surface of the surface of the endoplasmic reticulum becomes rough because of the presence of the ribosomes. So that is why they are called the rough endoplasmic reticulum. Clear? Please go through the functions also, the smooth and rough one. So in case of this is also related to protein synthesis, and it's also because ribosomes are attached to it. So the main function is protein synthesis obviously, and also certain various maintenance and the mineral balance and etc. Next one what we go is a very nice structure, and what you can say is a very fascinating structure, that is the lysosomes. Okay, these are also called the suicidal bags. Yes, you have heard it right, it's also called the suicidal bags. So powerhouse was mitochondria. Now suicidal bags are called the lysosomes. Why? Because lysosomes are granular structures present in this cell, and they have the capacity to release enzymes. Okay, that enzyme, that is the digestive, they release digestive enzymes. Now this digestive enzymes sometimes what happens, go against the cell only, and what it does, it actually digests the cell itself. So when it is going against it, it's sometimes digesting the cell itself. So that is why it is also called the suicidal bags of the cell, because lysosomes target to release enzymes. Enzymes, and these enzymes sometimes go against the body, and it actually definitely digests the cell only. So that is why it's called the suicidal bags. Okay, they are also present in different minute minimum numbers, and their main work is for secretion of digestive enzymes. Done. Next we come to the main part of this one. I hope I have cleared all. Golgi apparatus, vacuole, these are all outside external materials outside the nucleus, but when we will go inside the nucleus, they are other animals and other cell organelles. These are all organelles which are present in the cytoplasm. So let's revive once. Golgi apparatus, vacuole, cell membrane, mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum, and then ribosomes, then centrioles. Yes, I have missed one more there, the centrioles. This structure is centrioles, are X-like structure which you will not find in plant cell, but in animal cell it's very much present, and this actually infuse the determination of the the cell division and all those the centrioles. Actually it infuses cell division, and all the other reciprocating activities will be performed by the centrioles. So this is another structure, half X-like structure, which is present in the cytoplasm, and it helps in these type of cell division activities, which is not present in case of the plant cell. Right? Next we come to the, after this we have conclusions these things, we will hit the mastermind of the cell. Who is the mastermind or the controller of the cell? The nucleus is the main mastermind which is controlling the whole set of the nucleus. The first part will be your nuclear membrane. Again a question shoots out, that what was the need of placing another membrane inside the cell membrane? Because you have to differentiate whenever a membrane is allotted. Whenever a membrane is allotted, that means you want to separate two things separately. So that is why when cytoplasm is present and inside the materials which are inside the nucleus, that has to be separated from the cytoplasm, that time obviously you have to infuse a boundary. And in order to infuse a boundary, what do? You put a new membrane, that is called the nuclear membrane. This is also the main activity, is to separate out the other cytoplasmic structures from the nucleus and providing extra support, and obviously controlling the semi-permeability. This is not semi-permeable, this is actually a fusing structure which allows different items to get separated from the cytoplasmic structures. This is a nuclear membrane, and main function of the nuclear membrane is to provide support and rigidity to the cell. Next, what is it? Inside the nuclear membrane, you will find a type of structure that is a nucleoplasm. Where is it? Nucleoplasm. Here comes your nucleoplasm. Now children, see one thing. Cytoplasm. I have split it out. Cyto means cell and plasma means the material which is inside the cell. So that was cytoplasm. Now as it is inside this plasma-like substance, this plasma-like substance is inside the nuclear membrane. So we are only changing this plasma, we are keeping the same, but only where is it? The location is inside the nuclear membrane. So that is why we called it as a nucleoplasm. So if you split it up, you can see nucleo and plasm. Sorry, nucleoplasm, plasma. That means what? Nucleoplasm. Nucleus is this one and plasma is the material, so semi-solid material which is inside. This is the nucleoplasm. What is the main function? It is also providing a base to other nucleolus and other activities and maintaining the consistency of the cell. Next, after the nucleoplasm, we come to the next part. This is the nucleolus. So inside the nucleoplasm, so there are 3 to 4 things which are actually providing a project. Now to see, don't get confused. First is the nucleus. This total thing is the nucleus. But when you are separating out the nucleus, you will find the first part is the nuclear membrane. Second one is the nucleoplasm. And inside it there is another neutral structure. This is called the nucleolus. Inside the nucleolus, you have the chromatin network or the chromosomes. These chromosomes are actually carrying the genetic materials or the hereditary materials which actually determines the gene, our parental character and all the hereditary unit. The genetics is actually focused in this one. So this is what is the total structure of the animal cell. And these are the specific functions of the different cell organelles, and along with it, this is the nuclear membrane, nucleolus, and all the functions of the nuclear activity. Now what is the function of the nucleus? First one is it provides extra support, it controls, it is a master unit, it controls all the other activities of the cell, means controller of the whole cell, and it also determines the genetic or the hereditary materials present inside everyone's body. Clear? So this is a typical animal cell and typical animal cell organelles with their functions I have reviewed to you. So now what is your duty is to go through, pause the video, see whatever I have explained and go through each and every names and the spellings and try to split them up the way I have taught you, and just go through the functions. And functions must be in points. Never write functions in summary, write the functions in points. That creates more image of your knowledge to the teacher. Okay, hope you will like this video and enjoy this video. And while you have gone through these, and also go through the typical word, the common names like powerhouse, suicidal bag, nucleolus, who is the hereditary unit, master unit. These are other names, common names used for these type of cell organelles. Okay, semi-permeable, who is a semi-permeable membrane. So these are the common names which sometimes do come in your exam, and people often get confused that what is that. Okay, so please make a determined and clear idea of the animal cell. If you have any doubts, please do make me a comment in the comment box section. I will try to clear out all your doubts, and next video I will upload on a typical animal plant cell. Okay, so please keep a follow up with my videos.